Alright, so we're going to continue on this chapter. So the next topic is DNA replication. So we all know that DNA can replicate itself. And remember, we talked about cell division. And in the S phase of interphase, this is where DNA replicates itself so that you can have two sets of genetic materials that will be distributed into the two new daughter cells. So there were different proposals as to how DNA will replicate itself. So before we know the correct answer, there were three hypotheses. Okay? So the first one is conservative. So in this proposal, some people hypothesize that the parental DNA remains together, and then the newly formed daughter strands are together. Okay, so this is the parental DNA, and then this is the new daughter DNA. So this is conservative replication. And then the second proposal is semi-conservative, so that you can see each new DNA molecule has one old strand and one new strand. So it's like half conservative, right? So this is why we call it semi-conservative. And then in the dispersive method, for each new DNA molecule, you will see that within the strand, you have old and new, old, new. So the new DNA kind of segments are dispersed, within the new DNA molecule. So it turned out, again, by a lot of experiments, we found out that the semi-conservative is the correct way, for DNA to replicate itself. Okay, so before we replicate the DNA, it's very important that you separate the two strands first, and then you can use each strand as the template. So each of the old strand will be used as the template, and then you use these templates to make a new DNA strand, right? So if this is the template strand, then the green represents the new DNA strand. All right, same thing here. If this is the template strand, the green is the new DNA strand. Now, here's my question. So how do you use the template strand to make the new strand and make sure that the two new DNA molecules are exactly the same as the parental DNA? So how would you do that? Right, through the complementary base pairing, right? So we're going to look at this very closely. So the second piece of information from this slide is the enzyme that will split or separate the two DNA strands. So that enzyme is called helicase. I remember ASE is usually an enzyme, and this enzyme is going to separate the DNA double helix, so it's called a helicase. Now I have this picture here. I borrowed it from another instructor. I thought it was funny. It's kind of like a dirty joke, but I think it's very funny, and then you will really remember this enzyme for the rest of your life. Now I know a lot of people like the Big Bang Theory, the TV show. So you probably know who this person is, right? He's kind of like a dork, and he likes to make kind of dirty jokes. So, he's also a scientist, right? So he said, I wish I was a DNA helicase so I could unzip your genes, right? So DNA is unzipped by the helicase. Okay, so you can really think of DNA, actually, the double strands as a zipper. And then when you unzip it, it kind of separates the two DNA strands. Okay, so that's the first step for DNA replication. So separate the two strands. And then you are going to have a series of enzymes that will participate in the DNA replication process. Okay? So you are going to have a primer. So we'll talk about these in more details. So a primer is a short fragment of RNA. Okay? So you are going to make a short fragment of RNA first, and then you can use that to initiate the synthesis of the new strand. Okay, so I'll talk about how this works later. And then the new strand is extended or produced or created by the DNA polymerase. Okay, so that's the main enzyme. That's the enzyme that will synthesize the new strand. All right. Now, for the two DNA strands, we're going to synthesize both strands, right? So we can make two new DNA molecules using the semi-conservative replication. Right? So this is going to be this old strand, this is going to be the other one. So you separate those two and you use each one as a template. So for those two old strands, template strands, we are going to have a leading strand. So this strand is synthesized continuously. The other strand, because of the direction thing, it's going to be called the lagging strand. And you cannot synthesize the new strand continuously if you use the lagging strand. Okay, so you have to synthesize the new strand in short stretches, and those stretches are called Okazaki fragments. Okay, so this is very important, and this is actually named by a Japanese scientist who discovered these fragments. And I believe that he won a Nobel Prize, which is, probably the most prestigious award that a scientist can ever get. Okay, let's look at the details, and then we'll talk about the last two. Alright, so first we're going to look at the DNA polymerase and the primer. Okay, so let's say this is the DNA strand, right? This is the template strand, specifically, the template strand. And then you're going to use the template strand to synthesize a new DNA strand, like this. And then this will be one of the DNA, the new DNA, okay? I'm just going to call it the daughter DNA to kind of differentiate this from the parental DNA. Alright, so the DNA polymerase let's say this is the DNA polymerase, like a little guy. So the DNA polymerase can grab the raw material. Okay, so you have a raw material. Let's say you have these different kinds of nucleotides. Okay, so the DNA polymerase can grab these raw materials, and then it's going to read the sequence of the template strand. So if this is going to be C, the DNA polymerase will grab a G at the end of the strand. Okay, so again, it's going to extend the new strand in this way, right? So the next one, the enzyme knows that, okay, I'm going to add A. And then again, it's going to read the template strand. If it's T, it's going to be A again. And if it's A, then the enzyme is going to add a T. Okay? So this is how the DNA polymerase can extend or synthesize the new strand. All right. Now you probably noticed that the DNA polymerase doesn't work from the very beginning, right? This is the beginning of the strand. It kind of starts after this short segment, which is called a primer. Okay, so the DNA polymerase really starts working from here. And that's because there is a limitation for DNA polymerase. It cannot make the new strand from nothing. Okay? So that means if this is the beginning of the new strand, if there's nothing here, the DNA polymerase cannot start from scratch. So it has to have a short fragment already created, and then use that as an anchor, and then it can extend the strand. Okay, so you have to have something that's already there for the DNA polymerase to start. So this little strand is called a primer. So the primer is actually produced or made by primase. So this is the enzyme that we didn't mention in the previous slides. So when it's ready to synthesize the new strand using this template strand, you are going to have a primase first to come here and lay down a few nucleotides, which we call primer. And then the DNA polymerase will come in and then take over, the job, start synthesizing the rest of the new DNA strand. Okay. Now, one thing you need to keep in mind is that this DNA primer, this primer for DNA synthesis is actually a fragment of RNA. Okay, it's not DNA. It's a fragment of RNA. Okay, so once you are done synthesizing the new strand, you can't have a little piece of RNA in the strand, right? So what you are going to do is, over here, the RNA primers are replaced with DNA nucleotides. Okay, so you will have enzymes coming here, they will remove the RNA primer, remove it, and then they will put down the nucleotides for DNA here. Remember the difference is whether it's a ribose or deoxyribose, right? So don't worry eventually this RNA primer will be replaced with the DNA nucleotides. So the entire strand is DNA and not RNA. Okay, so just to recap, something you really need to remember is that the DNA polymerase cannot start from nothing. It needs an anchor. So that anchor is called a primer. Okay, so that's the first important piece of information. And the second point is that you need to know this primer is actually a segment of RNA and not DNA. Okay? And then eventually this segment will be replaced by DNA. So that ensures that the new strand is completely DNA and not partially DNA, partially RNA. All right. The third point is that the primer is laid down by an enzyme called primase. Okay, so those are the three things that you need to remember. All right. So how does the DNA polymerase know in what order to add the nucleotides? So we talk about that, right? It's based on complementary base pairing. So if DNA polymerase reads this as an A, it will add a T in here to pair with the A. Okay, so this is how it works. All right, so let's look more closely at the process and introduce some of the new enzymes and some old enzymes. Okay, so this is the 2 DNA strand. Oops. So these are the 2 strands of the DNA that you want to replicate, right? So first of all, you need this topoisomerase to kind of straighten out the DNA double helix. Okay? So I have the function right here. So the DNA is really super coiled, right? So it's like, so think of, you guys may not know this, but the old telephone cords, you probably have seen that in TVs or movies. Remember the telephone cords are really kind of coiled, right? So DNA is kind of like that. So you need the topoisomerase to kind of relax the super coiled DNA, to kind of release all the tension from coiling. So that's what this enzyme does. Okay? Once you are done with that, you are going to have an enzyme that will unzip the DNA, right? Basically, it separates the 2 DNA strands. So do you remember what that enzyme is called? Helicase, right? It's right here. So the helicase is going to separate the 2 strands. All right. Now there is another protein that will come into work. So that's called the single strand binding protein. So once the helicase separates the 2 strands, they will have the tendency to come back and kind of bound again, kind of linked again. So you need some proteins to kind of keep the 2 strands going back to be linked. So that's what these proteins do. Okay, so that's why they're called single strand binding proteins. They will bind to the single strand and keep them as a single strand. Okay. So now that the 2 strands are separated, you are going to have the next enzyme to lay down the RNA primer. Okay, so what do you need? Which enzyme do you need? So that enzyme is called a primase, right? So the primase will lay down a short segment of RNA, and that short segment is called a primer. Okay? So you lay down the primer, and then now DNA polymerase can come into work because it can just start from the end of the primer and start adding the nucleotides to the growing strand. Okay? All right, now there's another enzyme I didn't talk about, and this is gonna be the DNA ligase. So the DNA ligase can connect the nucleotides together, can kind of link the nucleotides. Also, when you have, let's say this is the new strand, and then you have the primer, right? And eventually you're going to kind of remove the RNA primer and replace it with the DNA, right? Let's say this is a segment of DNA. So the ligase can also link these 2 segments together. Okay? So it basically kind of almost like it seals the gap, right? Connects things and seals the gap so that everything is linked together. Okay. Okay, so this is a quick summary of the functions of different enzymes. So please read these descriptions because I will probably have some test questions on the functions of these enzymes. All right. Okay, so this is how topoisomerase works, and you guys can read the description and look at the picture. I think the picture is pretty good. All right, now I want to focus on the Okazaki fragments. Okay, so when you look at the function here, you can see that DNA ligase also joins the Okazaki fragments together. Okay, so I'm going to show you how the Okazaki fragments work. And this can be really confusing, so just kind of bear with me. All right, now remember we said that the DNA strand, the double strands have directions, right? So one strand, if it goes from 5 to 3, the other strand will go from 3 to 5. So they're anti-parallel, right? They go the opposite directions. Okay. So the new DNA strand has to grow or has to be synthesized from 5 to 3 direction. This is super important. You can expect a question on this for sure on the test. All right, so this is how it works. Okay, so the dark blue, these are template strands, right, from the parental DNA. And then the light blue is going to be the newly synthesized strand, okay? And the red is going to be the primer. Okay. So the new DNA strand has to grow from 5 to 3. So that means for the first template strand, that goes from 3 to 5, is going to be the leading strand. Okay? Why is that the leading strand? Because you see this is the new DNA strand, right? And you can see it's being synthesized from 5 to 3 in this direction. Because that's the direction that DNA polymerase has to follow. It cannot go from 3 to 5. It has to go from 5 to 3. So this is a 5. And then the other direction is 3. So the DNA polymerase, the little enzyme, synthesizes the DNA from 5 to 3 in this direction. All right? So if that's the direction, that means the template strand is going to go from 3 to 5, right? You can see 3 here and then 5 over here. So it goes from 3 to 5. So the template strand that goes from 3 to 5 is the leading strand because it's complementary to the new DNA direction. Okay, so 3 to 5 is totally the opposite as 5 to 3. And then we know the new DNA strand has to go from 5 to 3. So they are complementary, they kind of match with each other, right? So for this leading strand, the DNA synthesis is continuous. So it will just keep going from 5 to 3 until the entire strand is replicated, all right? Okay, so that's the leading strand. Now, for the other old strand, so this is the direction. So this strand goes from 5 to 3, right? 5 to 3. Now, this wouldn't work for continuous DNA replication because if this goes from 5 to 3, the new strand cannot go this way. 5 to 3. That doesn't work, right? Because these two strands are not going to be anti-parallel anymore. So these two strands have to be anti-parallel. So if your template strand goes from 5 to 3, your new DNA strand has to be synthesized from the other direction, right? 5 here and then 3 here. So that means once the DNA is unzipped, the new DNA strand has to go from 5 to 3. Okay, see my drawing here, 5 to 3. So it cannot grow from 3 to 5 because that's not how DNA polymerase works. It has to go from 5 to 3. So what means the DNA polymerase has to jump ahead a little bit. So instead of starting from here, it has to jump ahead a little bit and then synthesize a fragment. Let me use this. So the DNA polymerase jump ahead. It's gonna come here and it's gonna synthesize from 5 to 3. That's the correct direction for the enzyme. So it's gonna make this fragment, all right? And then it's gonna jump ahead again, okay? And then synthesize the next fragment from 5 to 3. And then it's gonna jump ahead again, right? Synthesize the next fragment from 5 to 3. Then it's gonna jump again. So each time the DNA polymerase can only synthesize a fragment. So it's not continuous. It can only synthesize a fragment. And then these fragments, don't worry, they will be linked together to form a continuous strand. So who's gonna link them together? The DNA ligase. So the ligase is actually really important. It can link the fragments together, link nucleotides together. So it's a very useful enzyme. Okay, so this is how you produce Okazaki fragments. I know this can be a little bit confusing, so if you have questions, just let me know. Or you can find a lot of videos on YouTube to kind of get some animations on this process, which may help. Okay. All right. Proofreading by DNA polymerase. So we talk about that why we use DNA to store genetic information, right? Because the information can be copied using the complementary base pairing. And also that pairing will ensure that we don't have too many errors, right? If you just follow the rule A pairs with T, C pairs with G, you shouldn't make a lot of mistakes. But the enzymes are not perfect, right? So DNA polymerase will make mistakes. So to kind of minimize the error rate, the DNA polymerase has a function, a proofreading function. So as it synthesizes the new DNA strand, it can also proofreading and make sure that it doesn't make too many mistakes. So this is the proofreading function by DNA polymerase. So here we go. It can correct the errors in replication. Right? Now, if an incorrect base is accidentally added, the enzyme can make a cut at the phosphodiester bond and releases the wrong nucleotide and replace it with the correct one. So this is how it works. So over here, let me just draw something real quick. So if you have A, T, C, G, T, right? So the correct pairing is T, A, G, C, right? Let's say the polymerase accidentally puts a T here instead of C. When it does proofreading, right, it notices that, oh, this is not the correct nucleotide. So it's going to cut right here. So it's going to break the phosphodiester bond. Remember, the backbone of the DNA is connected, linked by the phosphodiester bond, right? So it's going to break this bond and cut the T off. And it's going to replace it with the correct nucleotide, which is a C. I'm sorry, keep doing the G. That looks like G. So it's going to replace it with a C. And then it's going to link the C with the neighboring nucleotides. So this is how proofreading works. Alright, there are some other mechanisms that you can correct the errors. And these mechanisms require different enzymes. Okay, so the first one is called a mismatch repair. So sometimes the proofreading may kind of overlook some of the mistakes, right? So for example, over here you have a wrong pairing, base pairing, T pairs with G, but the enzyme didn't kind of spot the error, so it stays. And now you can use the mismatch repair proteins, or actually enzymes. These enzymes will kind of, parole and check your DNA, and then they can detect this wrong base pairing because the structures or the width of the DNA will be different. Okay? And then the enzyme can take out the wrong base pairing and then kind of correct it. So once you correct the GT base pairing, you're going to have the correct base pairing. So this is how mismatch repair works. So any mismatch error will be detected and fixed. The second one is nucleotide excision. So actually I studied this a little bit in frogs when I was doing my PhD. So nucleotide excision, based on the name, that you can kind of take a guess that the enzymes have the ability to take out a segment that have the DNA damage. Take it out, it's like exercise it, and then you're gonna have the enzyme to put in the correct nucleotides based on the other strand. So usually this type of repair will focus or target thymine dimers. Okay? So usually thymine dimers is caused by UV light, by UV radiation. Right? This is kind of related to skin cancers. So when you're exposed to sunlight, a lot of sunlight, the UV can damage your DNA, which may lead to mutations that cause cancer, skin cancers. So when the UV radiation, which has a lot of energy, gets through your cell and get to your DNA, it can cause an abnormal structure. So it's gonna cause this, it's gonna link the two thymines together and cause a structural anomaly called the thymine dimers. Right? So this will totally mess up DNA replication, right? Because when DNA polymerase gets here, there's a dimer here, it doesn't know what to do. The structure is not correct, right? So it doesn't know what nucleotide that is supposed to kind of pair here. So that tends to cause mutations and then cancer. So the nucleotide excision enzymes can detect this type of DNA damage and then take out this little short segment and then replace it with the correct sequence. And so this is how it works. Now, if you have abnormal function in terms of nucleotide excision repair, you may be more susceptible to skin cancer. And so that has been studied a lot, that has been proved. So that's actually very, very interesting. So in my research, I exposed tadpoles to UV radiation, which I know wasn't a nice thing to do. But I was trying to study the DNA damage caused by UV radiation, and then the enzymes that the tadpoles had to repair this type of DNA damage. Okay, telomeres. So the telomeres are actually very interesting, and there are a lot of people who are studying telomeres, which is related to aging. Okay, so the telomeres have been kind of implied as a mechanism that causes aging. So if you can kind of slow down the shortening of chromosomes at the telomeres, then you can slow down aging. So that's a very, very interesting topic. Hold on a second. I have to stop real quick.